basically, I wanted to describe two very different projects. So that basically, one uses very intensive gene editing in a mammalian genome, and the other one uses very heavy synthesis on a prokaryotic genome. To see a little bit the two different, basically some of the things that we have been discussing, how they could be, basically how we apply them in this case. So first, one of the projects that we are working in the lab is basically in humanizing the pig genome. I just put a couple of slides of background. So basically, there is a huge lack of tissues for transplantation. I'm sure you have heard there is a very long waiting list for organs, also very long waiting list for cell therapies like beta isolates, even hepatocytes. So there's a big lack of tissues that are needed for transplantation, and the pig has been proposed as maybe a potential solution because it's similar in physiology, similar in size of human, and it's easy to breed and to grow. So, and also another important thing is that pig is becoming more and more an interesting model organism, especially for certain diseases like heart diseases, also for embryology studies. So, however, to use pig cells for humans has some challenges. Basically, we have big immunological barriers, and also there is some safety issues. So the pig genome contains a lot of endogenous viruses that they are active when put in contact with human cells. So yeah, and today I'll focus a little bit on this part because it's very gene editing oriented. So basically, how we did to remove these viruses from the pig genome using basically just Cas9. So what are these viruses? So basically, they are called porcine endogenous retroviruses. They are being endogenous retroviruses. They mean that they are vertical transferred. So from basically they are encoded in the genome, meaning that no matter how clean you breed your pigs, they will always be there. Because they are transmitted basically vertically through the genome. Actually, they are some of the most active retro elements into the genome. So this is like a phylogeny of the different retroviral elements of the pig genome. We see this very clear cluster. Some people, there is some controversy. Some people thought that this was a big threat in the past. Actually, it really stopped many clinical trials, and it really stopped many big clinical programs from Genzyme and others to use pig tissues for therapy. How are these distributed? So basically, normally it's a repetitive element. So in all pig genomes there are multiple copies. In the cell line that we were working has 62 copies, and they are very simple elements. They are composed only by 3 genes and some kind of LTRs. So these 3 genes, GAG, POL, and M, and some LTRs. So, and basically, the thing that we wanted to do is to inactivate all of them. So inactivate the 62 different copies. So and how we did that? So basically, we followed the same principle that we were discussing before. So we designed some gRNAs in the part that we wanted to induce non-homologous enjoining. In this case, we use 2 gRNAs. However, the efficiency was relatively similar if we use 1. It's better with 2, but it was not radically different. And then basically we use one of our favorite constructs, which is a transposase, the piggyback transposase that I mentioned before, to deliver the 2 gRNAs and Cas9. Okay? Just a couple of words on how we design these gRNAs, because when we work with repetitive regions, the risk of off-target is much higher because we are cutting many parts in the genome, and normally repeated regions have many things that are similar to them, and we could cause chromosome translocations because you are cutting multiple sites on the chromosome. So I think extra care is needed for repetitive regions. If some of you are working with line elements or whatever repetitive, I think it's good to keep this in mind. So what we did in this case is we used multiple genome alignment to identify the most conserved areas. And then basically we designed gRNAs. We just wrote a small computational program to basically design a gRNA that follows these 2 principles: maximum specificity and maximum comprehensiveness. Now, actually, you could do exactly this with, for example, CRISPR-seq or other programs. And then, yeah, this is how it looks like. So the 2G RNAs cut this very conserved area. Here I just want to highlight that these PERVs, they are not identical. They are very similar, but they contain different haplotypes. This is a tree that basically represents these different haplotypes. You said there is some variation on the PERV element. I'm just mentioning this because we'll use it when discussing the results. And this is how the experiment went. So we started by cloning our piggyback with Cas9 with the 2G RNAs. Just to remind, this piggyback is this transposase that will insert randomly into the genome. So basically, we transfected the cells with the piggyback. We started pyromycin selection. This piggyback contains a pyromycin selection gene. And then after the selection took place, we assume that this has been stably inserted into the genome. And then we start doxycycline induction. And then we just harvested cells at different points. And measured how much editing. And then as you see, the editing goes from low to high, and then it goes low. So then, at the point of maximum editing, we harvest the cells. This is a population of cells. So we have cells that are edited, cells that are not edited. And we used a tool that is very efficient to help enhancing the efficiency, which is FACS. So we use FACS to sort single clones. And then we realized that most of the clones had no editing, and a few of the clones had very, very high editing. So if we put this into representations more similar to what we discussed before, so here in the x-axis, as before, we have the genome loci, and in the y-axis, basically the different reads, and here in red, I am coding the deletions. So for example, this is the population. So basically, you see this area is deleted. However, this area, these ones are not deleted. This is deleted, this is not deleted. And then in purple, I'm representing different haplotypes. So just to make, since it's a repetitive region, unless we use this information, we don't know what we are looking. We don't know if it's copy 1, copy 2, or copy 33. This was in the population level, 36%. That is the average of this, even though this average comes from 0 and a few almost hundreds. And then when we separate the different clones, we have clones that have no indel at all, so they are just clean. This is just background. And then we have clones that are all edited. You see red from down to bottom. And then, yeah, just a little bit of discussion of why this was like that. We were a bit surprised to see this bi-modal distribution, but well, basically it's what it is. And yeah, basically we have some evidences that maybe there was a little bit of gene conversion. So actually, even though this was a non-homologous end joining driven experiment, probably being a repetitive region transformed this into a little bit template-based repair, because one of the broken parts probably was used as a template to repair the newly broken parts. So we also inspected if we had some SNPs that could indicate like a special a phenotype of like some, but however, we didn't see any, not from the RNA-seq point of view, not from the SNP point of view, any big obvious change that could basically explain this binomial distribution. And then another thing that I think is very simple but very important is like Cas9 expression. So especially when we are using such a construct, we cannot predict very well if the expression will be high or low, because it will randomly insert, and depending on the area of the genome. So the clones that had high expression, they had high editing. So it sounds simple, but actually in ourselves we didn't think on that in the beginning. So yeah, what I was saying about gene conversion, I think from the plots we can guess that there is gene conversion because certain types of indels, for example, this type of indel, we see it in different haplotypes, meaning that at least we see in different genome areas. This is very likely that this happened by copy-paste, so some kind of gene conversion. And actually, if we make the statistics, there is a huge overrepresentation of co-occurrence of certain indels. So you never find in two different clones the same indel, but you do find the same indel within the same clone. So there is a little bit of this copy-paste type of mechanism. And then the last thing about the highly edited clones normally express more Cas9 than the lowly edited clones. So, yeah.
When do you decide to stop the expression of Cas9?
Oh, actually, in this case, we really measured. So, for example, we did the experiment and we just measured how much cutting we were having. And basically, the day that we had more cutting, we selected the clone, and after, but we selected the population and separated the clones. And then we just measured how much Cas9 had the clones that had all the regions edited, and how much Cas9 had the ones that didn't have editing. And basically, there was a huge association between high Cas9 expression and high editing, and low, and low.
After selection, do you keep on producing?
Yeah.
You never stop?
We never stop. But since we use this system that randomly integrates the Cas9 in different parts of the genome, some clones, probably this construct landed in a part that's very open chromatin, and it expresses very well in these cases. However, in other cases, probably just by chance, it fell in places that there is less expression, that is more closed areas. And this had an influence on the... That's why it's one of the reasons that I really encourage when designing constructs for long experiments, especially if you are making an animal, to really be very cautious in introducing things to protect your construct that keep, for example, these chromatin protecting elements like insulators, because probably you could prevent this bias a little bit by protecting well your construct. So yeah, we also ran a little bit of simulation of all these non-homologous joining versus template-based repair. So we use this Markov model that you can have the different transitions between 1 edit, 2 edits, all the way to 62 edits. And basically, this has two components. One that you can, after a cut, you can relegate with mutations, so non-homologous joining. You can relegate with mutations, without mutations, so no editing at all. So if you cut and paste the same, no editing. Relegation, so you cut, but then you make a mistake when ligating, it's non-homologous joining, or cut and then use a template to repair, which is the HR or template-based repair. So if you run a simulation with the system that we used, this simulation basically, H is the template-based repair, M is the error-prone repair, then non-homologous joining, and the different case is the number of cuts. So I think it's quite clear that the biggest factor is the number of cuts, which also correlates with the high expression of Cas9. So yeah, it seems to be a key factor to make your clone express well Cas9 if you want a profound editing. And I'll just finish here. So then we tried to check if these edited cells were infectious. They were not, so we couldn't see the viruses. And then just one very last thing. Another important thing that we discussed before, but we'll discuss more in the practical sessions, is about the genome stability. When we do cuts, we can have collateral damage. One option to take into to be careful with the collateral damage is whole genome sequencing. I think that here basically what we did is just sequence the entire genome and then inspect the areas where have closest proximity to the cutting site. I mean, in sequence distance. I mean, for example, I think here we use up to 2 mismatches. So all the regions in the genome that had up to 2 mismatches to our gRNAs, we just check that they didn't have any cut. Here I'm representing red for cuts. And in this case, we only saw cuts in the direct site, so the positive control. Although I have to say this is the peak genome, it's not very well covered, so we may have some false negatives, but we didn't detect them. So I think probably I'll finish here. And then we can continue. Oh, do you have any question before?